So the field of liquid biopsy has evolved tremendously in the last decade. Our first paper on the prognostic value of enumeration of CTCs in breast cancer was back in 2004. Since then, of course, there have been a number of studies focusing on CTC enumeration, but also CTC biomarker. Just 3 years ago, we put together all the information available from CTC prospective studies, and we asked the question if CTC, in fact, not only is a prognostic marker, but could be a stratification factor in patients with stage 4 disease. And we asked with almost 2,000 patients data, and we saw that, in fact, this group of patients with more than 5 CTC that we classify as stage 4 aggressive, it actually behaves in an aggressive way independently of the subtype, for example, ER positive, HER2 positive, and triple negative, irrespective of the site of recurrence, and irrespective of the line of therapy. So it's a very important prognostic stratification. As I mentioned, there have been a number of studies looking at the HER2 expression. High HER2 expression seems to predict high HER2 in the tissue, so can represent HER2 expression, a reflex test for HER2 status on the tumor. There is an HER2 low that now is becoming more interest for a number of application ADCs, and this HER2 low instead behaves quite completely different. And our group, as well as others, are trying to identify which are the drivers of this group of HER2 low. And then, as everybody knows, we move from the CTC field to the cell-free DNA field, where now there is a possibility to identify the pattern of mutation when you use an NGS panel in the different subtypes. You can actually identify in breast cancer actionable mutation. It's very clear that PI3K mutations are now actionable with alpelisib being FDA approved. And there are a number of, very large number of studies on ESR, multiple variants, and ESR mutations as a potential therapeutic target. Many of the studies in this field of drug development are actually focusing on developing new SERDs or SERD that will affect particularly the population of patients with ESR mutants and specific variants of ESR mutants. So clearly the field has evolved, and of course we know not only cell-free DNA is useful in metastatic disease, but because it's very sensitive technology can be also applying this in the detection of MRD or even early detection in the future. We know that mammogram can be not very effective in patients that have dense breast. Even MRI in that case, in the large population, screening tests may benefit at some point of some of this technology. The only issue would be that sometimes the detection in the blood comes much earlier than detection in the imaging. With regard to the MRD, this is an area of major investigation at this point, meaning that patients with high risk of recurrence may have detectable molecular residual disease with appropriate technology, some of them tumor-informed, some others based not only on genomic but also methylation. So these are certainly going to change the way we practice. We get to monitor patients at high risk and potentially escalate or de-escalate therapies in the adjuvant setting for breast cancer, similar to what is happening, for example, for colorectal cancer.